Hello and welcome to the Weektree Online Academy. My name is Chris and I'll be your tutor for today. In our previous video we looked at questions 6 through 10, and in today's video we'll look at questions 11 through 15. This is for the Cambridge AS Level Biology paper 1-1 of winter 2019, course code 9700. Okay, question 11. Some foods contain hydrogenated vegetable oils. These are unsaturated fats that have been converted into saturated fats. Which properties have changed here? Well, you need to understand the chemical formula of saturated versus unsaturated. So the unsaturated would have the double bonds and the saturated would have single bonds. Okay, and if you remember that the double bond effectively can cause a kink in the chain. However, the saturated ones generally form quite a linear structure. Okay, so let's have a look at some of the options here. Well, let's have a look at some of the incorrect answers first. Well, it says they will remain at liquid at room temperature. Okay, well this isn't actually true. By the conversion of saturated fats, this effectively increases the melting point anyway. The reason why the unsaturated have a lower melting point is because of this kinking that takes place, which means there's less van der Waals forces between each chain of the corresponding one because they're effectively further away from each other. That's why they have a lower melting point. So the conversion from unsaturated to saturated actually increases the melting point, not decreases it. They will have more double bonds in their molecules. Well, no, they won't, because it involves the breaking of the double bond and adding an additional hydrogen to effectively make it saturated. Okay. Their solubility in water increases. Well, no, it doesn't, because fats by nature are hydrophobic and insoluble anyway. You haven't changed that chemical property, so it has nothing to do with solubility. This leaves only option A. Okay, their chains will fit closely together. Well, yes, so instead of having the chains kinking off in different directions as an unsaturated fat, since they're now in a linear kind of straight chain, they can effectively be more compacted together. So you could have chain after chain after chain after chain closer together, rather than all these kinks that have come off the unsaturated chain itself. Okay. Question 12. Which statements could be used to describe enzyme molecules and antibody molecules? So hydrogen bonds stabilize the structure of protein and are important to function efficiently. 2. Hydrophilic R groups point to the center of the molecule and cause it to curl into a spherical shape. 3. The tertiary structure of the protein molecule plays an important role in the functioning of the protein. So let's have a look to see which statement is true or false. Well, statement 1 is very true. The structure needs to be stabilized, particularly in the case of the enzyme and the antibody. The shape is very specific. So a particular enzyme will have a very specific active site for a substrate, and an antibody will have a very specific site for where the antigen will bind successfully to the antibody or not. So by changing that shape, it does inhibit its functionality and its efficiency. Okay, so the hydrogen bonding is very, very important. Statement 2 is false. Okay, if we remember, it is actually the hydrophobic R groups that's on the center of the proteins, okay, as they are water hating. The hydrophilic R groups will be located on the outside of the protein and will actually cause it to bend outwards rather than inwards, okay, into the spherical shape. Statement number 3, well, this is very true. The tertiary structure is very important based on the type of bonding that takes place there between the various amino acids and the R groups, okay? So statement 3 is very true. Now, if you could have eliminated option 2 as being incorrect, you could have eliminated option A, B, and D as your incorrect answers, leaving only option C as the correct answer. Question 13. The Vmax and Km are determined for an enzyme catalyzed reaction. What will the effects on the Vmax and Km in the presence of a competitive inhibitor? Okay, well, if you remember that Vmax is constant for particular enzymes, okay? Now, the only way to alter Vmax would be to either increase or decrease the concentration of enzymes, okay? And this question does not state that we're doing that. So Vmax stays the same unless you are altering the enzyme or the concentration of enzyme, okay, for a reaction. Now, the competitive inhibitor, what it does is that it binds to the enzymes to form enzyme inhibitor complexes. Now, in theory, what happens is that these enzymes effectively now become inactive and they aren't going to bond with the substrate. However, this does not change the overall affinity between enzyme and substrate. It only appears to influence the Km value, calling it the apparent Km value. Now, if we remember what Km was, it was referring to the amount of substrate required for enzymes to be working at effectively half speed as such. So because of that, there's now more substrate available, and because of the loss of the enzymes in the due to the inhibitor complexes, it means it requires more of the substrate to effectively work at half its running speed. Okay, now as a result, the only one that leads to an increase in Km is option D. Okay, therefore we can eliminate option C from our choices. The graph shows how concentration of components 1, 2, 3, and 4 of an enzyme catalyzed reaction changes with time. Which row identifies the components of this reaction? So we have the words, let's pick product, enzyme, substrate, complex, unbound enzyme, and substrate. Well, this is a pretty easy question, but it might look more challenging to start with. We know that substrate is high at the start of a reaction and product is low. But at the end of the reaction, you have a high product and a low substrate. And the one that fits in with this pattern is 1 shows the creation of a product. Okay, so product increases through time and substrate decreases through time. So number 3 is the substrate. Okay, we can figure out the answer from this piece of information because we can eliminate A and we can eliminate B straight away. Now, by knowing what 3 was, we can identify that 3 is substrate, giving us the correct answer of option D. However, if you wanted to just explain part 2 and 4, well, at the start of the reaction, unbound enzymes are high. Through time, what happens is that they successfully bind with the enzyme, which means there's a decrease in concentration of unbound enzyme. As there becomes more product and less substrate, there effectively leads to an increase in unbound enzymes because there's not enough substrate to fill the active sites. Okay, so 2 refers to the unbound enzyme. Okay, so at the start it's high, it decreases and then increases again at the end. Okay, an enzyme substrate complex is inversely proportional to this, the fact that to start with it's low and then through time it increases, and then as time goes on and there's less substrate, there's less enzyme substrate complexes, which leads to a decrease. So number 4 is the enzyme substrate complexes. Okay. Question 15, which statement suggests that a membrane protein is involved in active transport? Well, let's remember what active transport is for a second. It's the movement of substances from a region of low concentration to high concentration against the concentration gradient using energy that has been released from aerobic respiration. And it involves the use of carrier proteins. Okay, this is one which relates to the question. So it's carrier proteins is the main one, which means they have to be able to change shape. So let's have a look at the options. Option A. It allows movement of molecules across a membrane if a concentration difference exists. Well, this is not specific to active transport because diffusion can take place from a high to low concentration and it doesn't involve active transport. So this is not a clear indicator. Option B, it can only function if the mitochondria are supplied with sufficient oxygen. Well, this statement is definitely true. The idea is that we need energy released from aerobic respiration to supply the energy to the carrier proteins to be able to change their shape. So B is definitely a true option here. Let's just eliminate the other 2 here. So C, a tertiary structure with a binding site with a specific shape. Well, we're not talking about a receptor or an enzyme here, so the idea is that this is incorrect. It can be found on the cell surface membranes and the mitochondrial membranes. Well, this does not indicate anything about a membrane protein, it's just a location factor. Okay, so the only option here is B. Thank you for watching our video today. Please register on the website for a free demo, and I look forward to seeing you in the next video.